Okay, all right. So have the slides come back?
Yes, ma'am. It was always visible.
Okay, okay. All right. So now let's look at the marks distribution. So, in this, like our previous semesters, we are going to have a midterm exam, which will be for 25 marks. Then there is going to be a quiz, which will be either multiple choice question or you will have short question papers. And this will be after our midterm. Okay. And then there is also a term paper for 15 marks. There is a seminar which will be evaluated for 10 marks. And this seminar, as it shows here, will be based on the term paper which you have done. Okay, so please take notice of that whatever subject you are going to choose for the term paper, you will be asked to make a seminar presentation on the same subject. So you don't have to write a different on a different subject matter. And participation in class discussion will be for about 5 marks. And finally, the major will be 35 marks. Okay, so I hope all of you will carefully have a look at it later on when the slides are distributed, so that you are mentally prepared as to how you are going to be evaluated in the course. Now, attendance policy, we didn't have it mandatory earlier, but now, with everybody getting used to the online system of classes and all that, the minimum attendance which is required is 90%. Okay, so I'm going to call out students' name just to make sure that you are attentive in the class and you're also understanding. So besides your login, your attention to the class, your ability to answer very quickly when your name is called out is also going to be looked at. So our attendance for this, that is, you have to log in at least 90% of the time and be attentive. For audit, the minimum requirement is 40 marks out of 100. Okay, so unlike, for example, please take note, because this is a change from the previous years, that at institute level for many courses, they have an audit of 30, but then now the Senate has also allowed it to go up to 40. So the audit requirement for this course is going to be 40 out of 100. And the student should have also taken at least 2 out of 3 exams. And what are these 3 exams? As they are listed on the list, we are going to be conducting a minor, or we will call it a midterm exam now, a quiz and a major. So you must have taken at least 2 out of these 3 exams. And when it comes to logging in for your attendance, 75% attendance is mandatory. So I hope this is clear to everybody.
Yes, ma'am, it is clear.
Okay, okay. Now, other students, so one by one, would the students please come, because you see, this is an audit course. It's not a core course. So would people, just in terms of their roll call, if you remember where you are placed, or just one by one, just let me know what is your interest in environmental biotechnology and what do you understand about it. Okay, so please start, because you have access to how you are placed on the roll call. So just go according to that. Okay, so, I can't hear anybody. So please come back if you are logged in.
Ma'am, can I start then?
Please start, Himanshu.
Basically, so good morning, ma'am. Basically, I'm a fourth year undergraduate student. So I did BTech in biochemical engineering. So I was keen to learn how what are basically the uses, that the principle that we are going to apply the biotechnology in real life. And ecology, environment ecology is one thing where we could learn things. So that's why I took this departmental elective, because of my own interest. So I'm eager to learn about that.
Okay, fine, Himanshu. Deepak, I think your full name is perhaps Deepak Kumar. Are you there?
Good morning, ma'am. I took this course because I want to just learn that how we can use biotechnological approaches to solve the environmental issues. Like there are many ones like global warming or like decomposing the plastics and that kind of thing. So I was thinking to like learn about these things and then apply them in real life to solve problems.
Okay. And then Kumar Kaushik.
Yes, ma'am. Ma'am, I
So
took this.
Kumar, Kaushik, how do you like to be called? Because your first name, it shows as Kumar. So are you called as Kaushik?
Ma'am, there are two Kosik in our branch, so it would be better if you call me Kumar, ma'am.
Kumar. Okay, okay. So tell me, Kumar, what interests you in environmental biotechnology?
Ma'am, I took this course because, ma'am, environmental biotechnology is applied and used to study natural environment. So, by taking this course, I also able to know about how we use biotechnology in our environment and in our real life how to use to. What are the uses of environmental biotechnology? That's why, ma'am.
Okay. So are you aware of any examples where microbes are being used?
Yes, ma'am, but as of now, I didn't remember properly.
Okay, okay, all right. So Shresht, what about you?
Good morning, ma'am.
Good morning.
I took this course because I want to know about the uses of biotechnology in our environment, as as we have studied about the biotechnology in very nano parts, like in cells and other things. But I just want to learn about something that is happening in real world.
Ratnesh, how about you?
Ma'am, I was also just curious to know like how the biotechnology which we have learned till now, or maybe we will be learning in future, can be used for solving environmental problems like the global warming and many other things. Like we have a very for a very short amount of petroleum available, and that we know that we can solve by using by producing bioethanol and things like that, and waste water problems are there that we can solve. So that's why I'm taking this course.
Mhm. Okay, okay. Bhavya, how about you? Bhavya Agarwal?
Yes, ma'am. Like nowadays, the more and more focus is being on environment well-being. So I was just curious how biotechnology plays role. Since I know that biotechnology is one of the best way to deal with environmental problems, and I want to learn that.
Okay. And then we have Badawat. Badawat Prashant.
Hello, ma'am. Good morning.
Good morning.
Yeah, ma'am. Just I took this course because that how the environment problem solving by using of environmental biotechnology in both man and in ecosystem. That's it, ma'am.
Mm-hmm. Arish Khan, how about you?
Good morning, ma'am.
Good morning. You have to
Ma'am,
speak a little bit louder, please.
hello. Am I audible now?
Yeah, much better.
Yeah, ma'am. So, what I'm expecting is that this course will be addressing towards the problem, environmental problem, and like we'll be probably learning about renewable energy generation or biomass production, something like that, using biological processes or some problems that we are currently facing in day-to-day life due to environmental problems, and how we will be studying or solving those using biological processes. So that piqued my interest, and I took this department as elective.
Okay, Harshit.
Yes, ma'am. Am I audible?
Yes, yes, I can hear you.
Yeah, ma'am.
Go.
After doing multiple courses, I was realizing the importance and applications of biotechnology. Like it could be used in pharmaceutical, curing diseases, dairy products, and even in some other research like DNA fingerprinting and all. Then when I got to know about this course, I was curious to know then, okay, now this can be applied in environmental problems as well. Then I kind of Googled it while opting for this course, like what are the environmental problems which can be solved, like treating the wastewater and all. So that's when I decided, yes, I want to explore this side as well. Like, what are the problems that can be solved, and how do we, and how can we solve all those problems?
Okay, and Virendra?
Good morning, ma'am.
Virendra, yeah, please
To
go
be
on.
to be honest, I do not have any idea about this course. I took this course only because my friends told me to do so.
Yeah, but that should not be, at least now you should orient yourself to see what is the possibility and what kind of things are you capable of learning. So, all right, so let's get started. So today, what my idea is that we are just going to define, for although broadly everybody knows what is environmental biotechnology. So it is basically applying biological sciences or microorganisms or biological systems for solving environment related problems. Okay, we can't say that biotechnological routes are the only ones. Certainly, there are other chemical processes also which have been in operation for a long time, but gradually people have realized that applying chemicals, for example, or certain other methods for solving the pollution related problems leads to other kind of issues. There could be a toxicity which you are actually increasing in the process. So given all that scenario, today it is becoming increasingly clear that if we apply biological routes or microorganisms for solving the environment related problems, it is going to lead to greener technologies and far more environmental friendly technologies. That is the reason why people are trying to switch back to making use of microorganisms. Okay, so by definition, therefore, what we can say, like in the first line, it says that it is basically a combination of science and engineering disciplines. Okay, why do we say science? We say science because based on our knowledge of how microorganisms interact, how they metabolize certain compounds, their biochemistry, what kind of environmental pockets, for example, they apply in nature. We already know, for example, there are microbes which are able to grow in the wastewaters. We know there are microbes which are able to grow on petroleum products. And all that information is derived from science. Okay, because we have a solid microbiology background with us regarding microorganisms, their biochemical roots, their biochemistries, etc. And now we want to apply that, for example, in the engineering discipline, because based on our engineering knowledge in terms of how to develop a reactor, which is going to be favorable for the growth of these microorganisms, create conditions in a bioreactor which are going to be very conducive to growth of these microorganisms, so that now they can chew up various pollutants which are in the environment. Okay, so we must see environmental biotechnology as something which is going to be an amalgam or a combination of our knowledge of the basic scientific knowledge of microorganisms as well as the engineering disciplines. And only then the science can actually be translated into some kind of a technology. Okay, so therefore you must, and it's not in isolation, you should not think that it's only going to be only about learning microorganisms or what kind of, not another biochemistry course or something, but we will also see how reactors are going to be designed in order to apply, for example, different categories of these microorganisms for solving environment related problems. So environmental biotechnology, therefore, it involves use of either microorganisms, or there are products which could be derived from them. That is, it could be enzymes which are derived from these microorganisms and which is used for development of processes which are involved in, number one, prevention. Okay, so in environmental biotechnology, it is not all about curing. It is also about preventing pollution from happening. Okay, for example, when you say that we can use microorganisms in prevention of environmental pollution through development of green technologies. Okay, so in other words, what do we exactly mean by that? It means that, for example, it includes strategies to generate fuel from renewable resources. In other words, now you would like to lower the fossil fuel usage because microorganisms are capable of producing, let us say, electricity. Now, can we scale it up? Or you have heard about solar energy, for example, that of course, there are no microbes which are involved there, but photobioreactors, for example, where microorganisms can utilize solar energy like algae, for example, they can use sun's energy in order to build up lipids in their biological cells, cell walls. So algae are photosynthetic microorganisms and they can capture solar energy in order to build lipids in their cell walls. This lipid, all right, can then be converted into biodiesel, which is far more environment friendly. So that way we can also say that we have really prevented pollution from happening. Because if you were to burn fossil fuels, for example, then you will have all of these nitric oxides and you have these oxides of sulfur, which are sulfur trioxide, etc., which are released on combustion of fossil fuel. But now you are completely preventing that because you are going to produce biodiesel. Okay, so that is the reason why we use a term like prevention of environmental pollution. So is this term clear to everybody? So is it clear?
Yes, ma'am. Yes, ma'am. But my question was like, do we only have microorganisms or like we can use other things also?
Yeah, but the thing is then you will not use a term like biotechnology. You do have examples of solar cells, for example, where you have these photovoltaic cells which are going to capture solar energy and turn it into electricity or whatever. But then you will not use the term biotechnology.
No, ma'am. My question was that are there not other biological systems, like can't we use like some plant cells and use plants and other things? And
see, plants, look, plants can be used, but not for fuel generation. Plants can be used. Plants are very good, if you place them in an environment where the soil is polluted, they are capable of taking up many of these heavy metals like chromium, lead, arsenic, etc. They can be taken up by plant roots and then it starts to assimilate it in the body of the plant. So that way metal because that will be in the next category. Then you can use plants for bioremediation, like it shows in the next term. But in the first one, when you say prevention of environmental pollution, that means the fossil fuels, for example, will not have to be burnt anymore because you have biodiesel, which is a much cleaner fuel, or you have bioethanol, for instance. You can use microorganisms, certainly, for making alcohol.
Ma'am, for producing bioethanol, we can use other oils like mustard oil and maybe palm oil or jatropha oil and other things.
Well, they are all heavy oils. And actually, when you look at the alcohol technology, the alcohol is only, you have to look at the biochemistry behind it. Okay, sugar to alcohol conversion is a very well established process in all the cells, especially those cells which are growing under anaerobic conditions. They don't metabolize glucose completely into carbon dioxide and water, but they can convert glucose into their partially oxidized product. Like you can have ethanol, you can have acetone, you can have butanol, etc., which are all fuel molecules, or butanol, for example. So is that point clear? Because when you use the term fuel, all right, by fuel, what we are looking at, for example, are molecules like ethanol, which can only, biochemistry wise, it can only come from sugars. Sugar can be in microorganisms or whatever. There are pathways to break down sugar into alcohol or into butanol or into acetone, etc.
Yes, ma'am. But I guess like palm oil is used for production of bioethanol by some first producing palm oil and then using some enzymes or microorganisms for producing bioethanol, I guess.
Well, yeah, we will have to look into it because I'm not sure how lipids can be converted, because you see lipids, they are eventually broken down into fatty acids and acetyl coenzyme A. and acetyl coenzyme A directly enters into TCA cycle. It directly enters into TCA cycle. You cannot make alcohol. Anyway, we will, when we look at detailed pathways, then we will come back and address this problem. All right. So now,
Yes,
for now, I think we, in other words, in continuation to what I was trying to say, is that it is not only about treatment, but it is also prevention of the environmental pollution through using biotechnological routes. And then we look at the second one, that is bioremediation. That is when we can use microbes, either bacteria or fungi or any such kind of, or even plants, for example, when we can use it for treatment of polluted environments. Okay, that is to say, you could have solid wastes like it shows in the slide here, or you could have the liquid wastes which we are generating particularly in the water bodies. We all talk about Ganga being polluted, or you talk about leather industry waste, you talk about textile industry waste, or so many other, the painting industry, heavy metals and other things are leached out through the painting industry. So all of that, for example, will come under the liquid wastes, where we can apply biotechnological routes for bioremediation. It could be gaseous wastes also, like ozone, for example, or carbon dioxide. Excess of carbon dioxide, for example, is also something which you want to prevent. And there are microorganisms which can sequester this carbon dioxide by the use of their enzyme and convert it into something else. So CO2 is a gaseous waste, etc. Or you could have soils which are contaminated, which are petroleum wastes, etc. So in other words, we are trying to put all of this treatment technologies under bioremediation, and that is why it is classified separately from the prevention category, which I stated earlier. And finally, we can also use biological systems for what is called as monitoring, or we say biomonitoring of the environment and treatment processes. Why? Because many of these small microorganisms are quite capable of detecting very small levels of pollutants in the atmosphere. Okay, and we can use these biological circuits, or as I will explain when we get to that, even in small, even in traces of, if you have ppb quantities of certain metal ions, these microorganisms can serve as sensors, and they are going to light up, they will become fluorescent. Okay, so imagine if you have a biosensor, it is able to detect, or when you say biomonitoring, that means, it could be a wastewater plant, for example, and you would like to monitor at stage 1 or at stage 2 with respect to time, what are the changes which are going to be occurring in this wastewater reactor. And we can monitor it using biological systems, etc. So biosensors, biomonitoring devices, etc., all of those will be categorized under biomonitoring. Okay, so I hope all of these things are clear to everybody.
Yes, ma'am.
So please, students, other than Ratnesh, also I would like students to come forward and please be alert and attentive, because today it's a very, very fascinating subject. That go on.
Ma'am, are we going to learn biosensors as well, as you just gave the example about the technological?
Absolutely. We will also learn about biosensors. Now, except that, in biosensors, we are going to utilize a microorganism, which is going to sense the presence of something, transduce it, and finally there is going to be a signal. That signal could be fluorescence phenomena. So you will see if there is a metal which is present now in the water bodies, and you have a microorganism based sensor, then it is going to fluoresce. And you know that there are traces of arsenic, there are traces of lead, which are going to be in the water bodies. Okay, so is that clear?
And ma'am, are we going to learn about the designing of bioreactors, the complete designing and the calculation involved?
Well, I will only give you the final picture. What are the components? But then the bioreactor design is a course which you are going to be doing with Professor Sri Krishnan. Bioreactor design and operation. I don't know if it is running in parallel to this course, but he is certainly going to discuss a whole lot about these wastewater reactors which have been set up. And I will only refer to it in terms of what is expected out of this in relation to the microbial systems which we are going to be using. Okay, so I hope that part is clear to everybody. Because there are so many batch, you will have the sequencing batch reactors, and then you have the anaerobic reactors, etc., which are all used either in environmental in wastewater treatment plants or elsewhere. And I will only, I can only show you the design part in terms of how it should be suitable for catering to a microbiological system. Because you are not dealing with inorganic catalysts. When you deal with microorganisms, you have to make sure you're going to keep them happy and healthy. They should be able to metabolize, they should have a proper temperature, and they can only operate within a certain range of temperatures or dissolved oxygen, so on and so forth. Or the kind of things which you are going to feed into the bioreactor are going to be very limited, because microorganisms, there is only so much of COD load that they are capable of bearing. So in relation to that, we will discuss. So does that answer your question?
Yes, ma'am, it answers. And ma'am,
Okay.
one more thing I just want to curious about to know, that what are the engineering aspect? Is there some other design aspect apart from biosensors? Could you just clarify in this?
Well, in terms of, you see now, when you, like, for example, take the example of wastewater treatment plants. There is a phase, what is the conventional process is the aerobic sludge reactor, okay, where microorganisms are added. And but then what should be the constitution of that? And today, apart from removing the carbons, which is actually coming in the form of carbohydrates in the wastewater plants, there is also a problem of nitrogen, because remember, all the food wastes that you are dealing with, the food wastes not only contain carbohydrates, but they also contain proteins. They also contain lipids. And when proteins are hydrolyzed, there are nitrogen containing compounds which are going to be generated. Like, for example, all your ammonia and everything, nitrogen containing things are going to be there, which, if not treated in a wastewater treatment plants, leads to release of nitrogen in the water bodies. Okay, and that causes growth of algae, because all algae need is a source of, it has sunlight, it has carbon dioxide, and a lot of nitrogen, which, if it is not treated in the wastewater bodies, can lead to what is called as algal bloom. And then you will see a green layer which is floating on top of these river bodies. And that is also very bad. And that is called as algal bloom. I think you must have seen, many of you, जिसको हम काई भी बोलते हैं हिंदी में. You must have seen that where the water bodies, the ponds or the other water bodies are polluted, you will see a green layer on top of it. These are basically algae which are growing in an environment which is very rich in nitrogen. Okay, so getting back to where I started, apart from treating the carbon, the COD, which is in our wastewater streams, we also need to treat the nitrogen. And for that, our reactors are going to be nitrification, and there are going to be components of our activated sludge process, like nitrification, denitrification will be one reactor, which is followed by the activated sludge process reactor. Okay, so in other words, when we even decide to plan a reactor for the treatment of wastewaters, we have to take the problem of removal of nitrogen, which is done through a nitrification denitrification reactor, which is then followed by activated sludge process. Or you could have activated sludge process, which is then followed by nitrification denitrification reactor to take care of that nitrogen, which is otherwise going to be released into the water bodies. So does that answer your question? All right. So So I hope that answers, because when you say the design of reactor, of course, Professor Sri Krishnan does. All right. But then what are the necessities? What are the components which need to be there in order to address in toto the problem of wastewaters will involve a relook at all of those systems. So should we move to the next slide?
Ma'am, I had a question, like, just.
Yeah.
Ma'am, like, I was reading in the news that Japanese scientists have developed some masks which detect coronavirus. Like, it will start glowing when some virus is there on the mask. So, we can consider that
Yes.
as a biosensor, right?
Absolutely, absolutely. Because you see what they are trying to do, although I have not read about it, but I'm just taking a guess, is that one of the core proteins of this coronavirus, okay, is what they call as a spike protein. Okay, so the coronavirus on the outside, you will see that it has some of these spikes, which are emerging out of the surface of the virus. Okay, now imagine that if that spike protein has luciferin, or it has a gene which codes for luciferin protein, and which is luminescent. Okay, so if this gene is, for example, replicated, or if it is in large quantities, okay, at which the viral load is very, very high, it is certainly going to be linked to something else which will allow at genetic level for it to glow, for instance. So if the concentration of that protein builds up to a certain level, then it turns on something else which will activate the luciferin protein, and you will see bioluminescence. Okay, so all of these can be embedded in the mask itself. It is not impossible at all, because what you are trying to judge in the mask is the viral load. So if this viral load, that means if there are maybe 1000 particles of the viruses, that spike protein is also going to be produced in large quantities. And what you need is a microorganism which is going to be capable of detecting these 1000 molecules of the spike protein. Once it does that, it just sends out a flash of light. So that is a kind of technology which you can develop, and that is nothing but a biosensor. Or it could be something like a biomonitor, because you are also able to monitor now what is the viral load in the atmosphere.
Yes, ma'am, that is possible. Ma'am, there was another example that a PhD student is there, like he's from electrical engineering department. So I talked to him. So he was talking that he was working on developing RNA thermometer. So that RNA thermometer, that is related to biological systems. So that can also be considered as a biosensor, right?
Certainly, sir. You see, I tell you, because that is the reason why, if you look at, for example, the second point which we have here, okay, which is either microorganisms we are going to use or their products. This product could be either enzyme, it could be DNA. There are certainly DNA sensors now. Okay, because DNA molecule is double stranded. So if you put a single strand which is complementary to that, if the viral load is there, then you can hybridize, you can create a double stranded DNA molecule, and that can light up something. So whether it is the use of DNA, RNA molecules, whether these are proteins, whether these are enzymes, etc., all of them are really within the domain of environmental biotechnology. Okay. So let's move to the next. All right. So, okay. So now this is the next slide where, I'm sorry, there is construction activity going on outside. So you can hear this noise in the background, but I hope my voice is audible in spite of this. Are students able to hear me properly?
Yes, ma'am, we are able to hear.
Yes, ma'am.
So there is a lot of noise in the background. So see, I'm just going to close the class in another 10-15 minutes because today is just an introductory class. So now, before we close, now all along I have been talking about, for example, the role of microbial systems, because at the heart of environmental biotechnology is the use of these microbes. Okay, now essentially we are going to be looking at two different categories of these microbial systems. The first category, which is shown in the upper picture, are going to be either the bacteria or they are going to be the archaea. Okay, now so there are some pictures which are also shown. Bacteria can be either rod shaped or they can be, for example, cocci, etc., or they can be spirals, whatever. They have different kind of shapes. And so these are, that is, the bacterial and the archaeal group are some of the most active biological systems. They are very useful because they have fast doubling times. Okay, now because if you are talking about environmental pollution or whatever, you certainly want to have a biological system which is able to grow very rapidly. You don't want very slow growing microorganisms, because the more the number of these microorganisms is going to be, the more rapidly they are going to be able to eliminate that particular pollutant from the environment. So these systems, microbial systems, should have very fast doubling times. Okay, and that is the reason why bacteria are preferred, because they have very rapid doubling times. Within 30 to 40 minutes, they are able to double themselves. And bacteria, what is the other feature of them, is that they have a range of metabolic activities. So when we try to investigate what kind of pathways do the microorganisms have, generally you will see that the bacteria are more versatile. Okay, that is, not only can they break down glucose into carbon dioxide and water, but there are groups of microorganisms which can break down, for example, glucose into acetone and butanol, or into alcohol, or into, or they can take up organic compounds from the environment. They can take up petroleum products, etc. They can even metabolize them to carbon dioxide and water. Okay, and that is what we mean by a range of metabolic activities. And that is the reason why bacterial systems are preferred. Okay, so I hope this point is clear to everybody that, most of the technologies or whatever people try to use the bacteria, they have very fast doubling rates. Then they have a range of these metabolic activities, which makes them very, very versatile. And they're extremely attractive, therefore. So some examples I have listed here, that amongst the bacteria, these are very ideal systems for biodegradation of organic matter, which could be, your all your hydrocarbons and everything, your naphthalenes and your aromatic compounds which are relieved or which are petroleum bioproducts. These are excellent for biodegradation of that kind of an organic matter. They are used in all areas of environmental biotechnology, and there is a constant search made in new ecospheres to identify more potent prokaryotic systems. You will recall that both bacteria, and archaea, they belong to prokaryotic systems. Okay, by prokaryote, what we mean that, their mitochondria or whatever, they are not well defined. There are no organelles inside, although they do perform the same kind of a function that a mitochondria does, except that they don't have these defined organelles. They don't have a defined nucleus, etc. It is just high school, your biology. I'm just trying to bring it back to you people so that you will remember when I use the term prokaryotic systems, that is, they don't have well-defined nucleus and mitochondria or whatever. Okay, so when you say that there is a constant search made in the ecosphere, by ecosphere, what we mean is, for example, hot springs or very, very cold climate like our poles, where the temperatures are very low, and yet there are microbes which survive there. Okay, or when you look at hot springs where the temperatures are like 160 degrees, 200 degrees, there are thermophilic microorganisms which are surviving. Okay, so today the stress is also to be able to go to these locations and to see what kind of life forms exists there and whether we can bring them here and we can use them for mitigating any one of these other problems which we have talked about related to the environment. Then we have archaea, which also belong to prokaryotic systems. And these archaea are particularly important because they operate in extreme conditions. That is extremes of pH. You could have very acidic soil or you could have highly alkaline soil, for example, and where even the bacteria cannot live. All right, but archaea are found to live in these extreme environments. Okay, or very hot springs, for example, you may not find a bacterium there, but you will certainly find some archaea there, which are life forms. Okay, so therefore, getting back to this point, that archaea are very good biological systems because they provide a very rich resource which will allow you to look for enzymes or other products which can operate under extreme conditions, and therefore you can set up processes where high temperature occurs spontaneously, such as fermentation, wastewater treatment, for example, because as metabolic activities goes on inside a wastewater treatment plant or during composting, for example, okay, then the temperature also builds up in that system, in which the other mesophilic organisms which we are normally familiar with, they cannot grow in that environment. So at that time, it is better to prepare a consortium which contains an archaea because it can sustain it can sustain that high temperature. Okay, so please try to appreciate, therefore, that what is the need to look for a resource, a microbial resource which belongs to the archaeal group. Now, the second groups of microbial systems which you are going to be learning about are, for example, algae. Now, these systems, they assimilate organic substrates very much like a bacterium, and they also assimilate carbon dioxide. That is, they can sequester carbon dioxide. Why is it important in environmental biotechnology? Because, we have disturbed the carbon cycle. We are producing a whole lot more carbon dioxide than is getting consumed by the plant life, or by the bacteria, by the photosynthetic bacteria, which are present in the water bodies. They are also capable of assimilating carbon dioxide. However, because we have disturbed this carbon cycle in nature, we have too much of carbon dioxide which is accumulating because of indiscriminate burning of fossil fuel. Okay, so algae, for example, are extremely good because they can assimilate light energy. These are photosynthetic systems, so they can capture carbon dioxide and they can assimilate it into lipids, like I said earlier, and we can use it, etc. For biodiesel formation, etc. But they can also sequester carbon dioxide. And removal of organic matter and nutrients, particularly nitrogen, etc. Because I had just given you an example that algae, all that they require is a source of carbon and a source of nitrogen. Okay, so the nitrogen or some of these nutrients which contain nitrogen can be completely taken away if you have algae, the picture of which is shown on the left, right here. Okay, so they can serve that kind of a purpose, but then one has to do it under controlled condition so that it doesn't happen in the water bodies, but it happens in localized ponds where wastewater can be brought in, for example, in localized water in the water bodies, and then algae can be made to grow there, and the treatment, etc., can be carried out. In addition to that, we also have these groups of organisms which are called as fungi. Okay, now fungi are very important in our ecosphere because on the ground when you see plant litter dropping in, it is mostly the fungi which are able to break down this complex lignin or complex organic matter into carbon dioxide and water. They basically grow on dead organic matter. Okay, so fungi, they degrade all categories of biopolymers like cellulose, lignin, agricultural residues. Sorry, what did you say?
I said that we were not able to hear you.
Your voice is breaking, ma'am.
Oh, is it better now?
Yes, ma'am, it's fine.
Yeah, maybe, because I'm also connected right now. We just shifted. I don't have a Wi-Fi, so I'm just using this mobile hotspot. Spot. So maybe, the connection is a little bit. So is it better now?
Yes, ma'am, it's fine. We got just 20 seconds of disconnect.
Oh, okay, okay. Doesn't matter. Doesn't matter. I hope it has come back now. I think next time it will be better because I will have Wi-Fi installed in the house. So anyway, going back to this particular part, algae, I had already explained to you that they are very important, although they have longer, because all of these organisms which are listed in this blue box are basically, these are all eukaryotic system, so they don't have very rapid doubling times. They take much longer to double themselves, but nevertheless these are very important. Algae, I had already explained to you that they can assimilate the light energy. They can also sequester atmospheric carbon dioxide because these are photosynthetic systems. They can remove organic matter, particularly that which contains the nitrogen or ammonia, and then assimilate it into biomass, etc. So imagine if you have an algae which can accumulate lipid in its cell wall, then it can assimilate all of these things, the carbon dioxide, the nitrogen, and convert it into a biological lipid which is accumulated in its cell wall, and that lipid can be used for biodiesel formation. Okay, so that is the reason why they are extensively being worked upon. And then the second category is that of fungi. So I have already explained that fungi usually grow on dead organic matter, and they are able to degrade very, very complex biopolymers. And not only biopolymer, but you will also see fungi capable of breaking down textile dyes, the aromatic compounds, etc. We are also working in the laboratory on some of these fungi. They produce these enzymes which can break down very, very complex aromatics. Okay, so because of that, they are used in soil bioremediation, particularly when you have petroleum things which are coming into the soil. We can use many of these fungi, and they are also being extensively used as biopesticides. Okay, now, if you use the other pesticides which are made through chemical processing industries, these are highly toxic. Okay, and now they are classified as environmental pollutants. So the idea is not to use those kind of pesticides which are coming from chemical industries, but people are trying to replace them using these biopesticides, which are nothing but fungal biomass. And they make certain kind of enzymes which are able to break down all of these, or they are able to remove many of these bacteria or whatever which are the soil pathogens which can otherwise attack your crop, etc. Okay, remember, pesticide is something to kill the pests. And what are these pests? These are the pests which can otherwise attack agricultural crops. So please put it in the correct context to understand. So pesticides are something which are going to kill pests which are infecting our agricultural crops. So you don't want to use any one of those things, chemical pesticides, because eventually they will end up in water bodies. They get washed out, they keep on getting accumulated in our water bodies, and it causes a pollution. But when you use a biopesticide like fungi, they can also inhibit these pests, and at the same time, this is a renewable biomass which will decompose automatically. The biological matter will return and get disseminated into carbon, nitrogen, and whatever. So there is no end product accumulation there of a biomass. Okay, so I hope you can understand what is the meaning of a biopesticide, and fungi can also be used for absorbing heavy metals. Because where are these metals coming from? As we will learn from this entire paint industry, which is a huge industry worldwide. Okay, or your batteries and everything, they all contain heavy metals. And fungi are capable of absorbing these metals, accumulating them in their biomass. So these are extremely important groups of organisms. And then we look at protozoa. And protozoa
I have a question.
Yeah.
Ma'am, I was asking, like, in algae, in case of algae, it accumulates lipids. So do they accumulate lipids only in their cell wall, or they can accumulate it inside the protoplasm also?
Well, I'm not sure. I will have to go back and look. Okay, because maybe they accumulate it inside. I know there is rhodotorula, and there are many other yeasts also which accumulate these lipids inside their cytoplasm. But as far as the algae are concerned, okay, algae, I think, can accumulate it in there because, biological membranes and everything are made up of lipids. So they will accumulate them. I'm not sure if algae can accumulate all of these lipids in their cytoplasm. I'll have to go back and see. I don't work on them. I have not read much about them, but I know they can certainly accumulate lipids in their biological membrane.
Ma'am, in case if they are accumulating only in their cell wall, it will not be much it in case if it is if they are accumulating inside the cell also, then it will be more effective, like, I guess.
No, you see, any part today, you have a way of breaking the cells and extracting lipids out through organic solvents. So imagine if there is an algal extract, okay, you just homogenize algae in a blender or something, and then you can add an organic solvent to it, carbon tetrachloride, or you can have anything else. Any organic solvent can be used to extract preferentially lipids out. It's not a very difficult process. Just a one-step process of extraction. Whether it is in the cytoplasm, whether it is in the biological membrane, doesn't really matter, because once you break the cells, as I said, extraction can be carried out by adding an organic solvent to it.
Yes, ma'am.
Okay, so
again,
that should not be. Yeah. Anybody else? Anybody else has a question regarding this?
Ma'am, I have a question regarding fungi. Like, fungi are they are not moving like animals, and they don't eat from everywhere and accumulate inside them. Like they are only grow where they are. So how can they like absorb heavy metals and enrich in heavy metals inside them? Like, when in case we want to heavy metals to be absorbed by fungi and extract it from fungi, we want the concentration of heavy metals to be more inside the fungi than it is there in the outside environment. So how is it possible? Like,
yeah. No, they are capable of assimilating. You see, for heavy metals, etc., of course, fungi are there. They will bioaccumulate. So imagine in a fungal cell, by weight percent, normally the metal concentration would be very, very low, but they are capable of accumulating metal inside. And then there are other ways of breaking these fungi open and extracting that bioleaching, etc., is also another process. So when we come back to metal remediation, that time we will take specific case studies and you will understand what is the biochemistry. Okay. So till then, I think we will, because all of these, whenever we take these examples, then you will also see the cases. I will link it with the case study. And then finally, we have protozoa, which is in the last category, which is also a group of, these are unicellular eukaryotes, which are in the wastewater treatment plant. Okay, so with this, what I'm going to do, I'm going to close today's lecture, and then we will come back on Thursday and continue. Okay, so please, I will share this particular PPT. So my only request will be that go through these 5 or 6 slides, which we have discussed in the class today, before we come back again, because then we will start with ecosystems and other things. Okay.
Yes, ma'am.
All right, so then I'll see you on Thursday.